Okay, so let's talk about gene regulation and expression. So, first off, we have a lot of genes in our body, and of our 23 pairs of chromosomes or 46 pairs of chromosomes, and all the genes that we do have, we don't know exactly why some genes are expressed and why some genes are not, and why certain people express some genes, why other people do not, is still not completely understood for every single gene that we have. Even though we have what we call the Human Genome Project completed, which has mapped out every single piece of DNA within the human body, we still haven't sifted through all of the information. Here's what we do know. Although we cannot always understand under why some conditions different genes are expressed, why others are not, one benefit to gene regulation is it becomes really effective. Remember that our genes are kind of like an expression of ourselves. We have to go through the central dogma always. DNA to RNA to protein. So transcription to translation. This is what controls our gene regulation. And these molecules can be expressed in what we call gene switches. So they can be turned on and off like a light switch. We can turn on a gene and we can turn off a gene. We can regulate genes in prokaryotes a lot better than we can in eukaryotes, just for that. So here's what we know about prokaryotes. 1, no nucleus, no membrane-bound organelles. So they have a single loop of DNA. This is really easy to manipulate and for us to understand. So they have what we call an operon. This is a gene that regulates the adjustment of DNA segments and controls which ones can be expressed and which ones cannot be expressed. The operons are common in bacteria and uncommon in things like eukaryotes, us. Bacteria, again, can be directly influenced by its environment, the food that they eat, the temperature of the environment. So they have to maintain an internal homeostasis or stable internal environment. And so they express their genes to do that. And sometimes this will change if they have a protein or if they don't have a protein being created. And with this, bacteria can flip on and off gene expression. So the most common example is a lac operon. A lac operon is found in E. coli, Russia E. coli. By the way, this is found in your digestive tract. And so we do have bacteria that is located within our small intestine that is extremely valuable for us because it allows for us to break down things that we normally couldn't break down. And also this healthy internal bacteria flora is really important for us to just be a normal, healthy person. So bacteria, not all bad. However, the bacteria in your digestive tract, if would happen to get out into the rest of your body, that could cause a lot of bad things to happen to you. So really bad. Anyway, Escherichia coli is in your gut and it breaks down lactose, the sugar found in milk. So we call it the lac operon because we're actually making, E. coli is making a protein that can break down lactose. So it makes a protein called lactase. ASE, protein. OSE, sugar. So let's look at this a little bit closer so that we can see. So remember, we are going to talk about the lac operon. This is the gene expression to make lactose. So let's see what's happening. Okay, so first your body has to take in lactose. If you don't have lactose, it won't go through this process. PS, and by the way. And so what happens when this lactose starts coming in, a repressor, it binds to this repressor. The repressor is like, no, what's going on? And then it signals the RNA polymerase to bring in the mRNA. Oh, wait, that's going through transcription and translation. And then what happens is that it actually produces something that we call lactase. And this lactase is really important because if we didn't produce this lactase, these enzymes wouldn't be able to break down lactose, and then you become what we know as lactose intolerant. Have we heard some of that before? So if you don't drink milk, or if you go on like 2 years where you don't drink milk, you become lactose intolerant because you lose the ability to produce lactase, or the Escherichia coli in your digestive tract lose the ability to produce lactase. One thing that's really important when we start controlling these is that we start looking at prokaryotes versus eukaryotes. Prokaryotes will have what we call a promoter site, and then it has an enhancer site. And so it brings those together. So gene expression in prokaryotes is a little bit more regulated than in eukaryotes. Obviously, we produce a lot more proteins. We have a lot more DNA. We have a lot more chromosomes. We have a big mess. So we have multiple factors that will trigger that gene regulation because we don't want an excess protein that we don't need. Such as if you don't have any sugar in your system, we really don't want to produce insulin. It would be really bad for you. Your blood sugars would drop extremely low. Your liver would freak out and flood glycogen all through your body. There's a lot of things that could happen. So we need these regulations to occur with a little bit of understanding. So we have these proteins that will help to regulate transcription and translation, which means we don't want them going on if they're not supposed to. These proteins are called transcription factors. They pretty much like switch it on or switch it off. Transcription factors influence not only if transcription can occur, but the rate at which it can occur. And we have some factors that are known also as repressors. Guess what? You're repressing something, stopping it. We also have enhancers. These are located on thousands of bases away from a promoter site. A promoter site is where we're actually going to start, or transcription begins is at that promoter site. That's where your RNA polymerase is binding, is the promoter site. So why would we have an enhancer near a promoter site? Well, what happens is an activator will grab onto that enhancer and it actually closes a loop. So it actually takes a segment out and allows for it to code faster. And the reason for this is because we have coding and non-coding segments of DNA. We don't know why we have these things called introns and exons. So when we talk about eukaryotes and all of our wonderful stuff, we have these exons which code for the amino acids and the proteins, and then we have them broken apart by introns. And we don't know why some people have more introns than others, and we don't know why introns were there, but we're sure evolutionary, they're really important. And so we can create splicing or variation over time, so we can change the introns and exons through mutations, and this creates a different protein. And so let's look what this means. So if we look here, our introns are broken up by our exons. Through transcription, we go and we have all of our RNA here. Our mRNA, though, is only made of our exons, no introns, because they don't code for anything. They're not important. From there, translation can happen, and we make our amino acid chain, which then we take and change that and modify it to make our protein. We just change the protein structure. Really important, these introns, these red segments right here, they're non-coding. And so when we're looking for these genes through the genome, sometimes it's hard to understand which ones are which because we have so many non-coding segments, and you don't really know they're non-coding until you go through transcription and translation. And if they're not found in translation, then you know they're non-coding. And so we're still trying to figure out this process. And so it's not always easy to identify exactly what gene is there. And that's why proteins through gene therapy are hard also in some cases, because we just don't know why they code for others and some don't. Until next time, when we talk about genome interaction.